last time we left off, we finished talking like collision theory and how it relates to orientation factor, right? That's the pre-exponential part of the Arrhenius equation. Some things need to collide in a particular orientation. So then the number of, well, actually, let me back up just a little bit farther. Right, so the pieces of the Arrhenius equation here, there's this pre-exponential factor that's A, and that's based on the number of collisions that are happening in whatever flask or whatever you've got. And usually that's fairly constant. But the thing that can affect that is the orientation factor. So sometimes you get fewer collisions or fewer successful collisions than you do total collisions in this equation, if I phrase that correctly. Yeah, fewer successful collisions or fewer successful collisions than the total number of collisions. And then the exponential part of it, that's where the activation energy is contained. Right? So increasing activation energy makes that exponent a larger negative number, decreases the chance of a successful reaction, but increasing temperature increases the energy of all of your atoms and molecules and increases the chance that they have enough energy on any collision that's in the correct orientation. All right, so that's where we left off. One of the cool things that you can do, and actually that quote that was on the very first slide about kinetics all being this very romantic dance between atoms and molecules, that guy won the Nobel Prize for using rates and kinetics to determine real, or at least to come up with valid models of reaction mechanisms. So reaction mechanisms are what is really happening inside any reaction, because most of them occur in several steps. So it's not just what we see as the overall reaction, right? Because the overall reaction is we put these two in the flask, and then this is what comes out. There might be, can be, and very often are, intermediate steps. So something else happens first, that creates an intermediate, and then that produces the products. So a reaction mechanism is the series of individual chemical steps by which the chemical reaction occurs, and then each elementary step in a reaction mechanism represents the exact species that are colliding. So what two collisions, what collision exactly has to take place to create the products, or to create an intermediate in the steps. So we're going to be looking at a lot of elementary steps. So for this reaction of, let me go back to this actually, hydrogen with iodine monochloride, this is the overall reaction, but it actually occurs in two steps. In the first step, hydrogen and iodine monochloride do react, and they produce hydroiodic acid and hydrochloric acid. Then the hydrochloric acid and the hydroiodic acid react in the second step to make the final products that we see, which will be the hydrochloric and iodine. So these individual steps will add up to the overall reaction. What I mean by that is, if we look at either side, if we look at either side of this reaction, we can see that on the right here we have hydroiodic acid, but we also have it as a reactant for the second step. So any hydroiodic acid that's getting produced in that first step then gets used in the second step. And we can look at those and say, oh, these cancel out, because it's not going to be part of the final reaction, and it's not one of the reactants that we put into the flask. Other things that aren't mirrored on either side in different steps will add together. So here the hydroiodic acid cancels out, and then we have H2 as a reactant, and then because there's iodine monochloride there and here, we'll get 2 of those. So they add together. And then our products, because we have hydrochloric acid plus another hydrochloric acid, that'll be 2HCl plus I2. So we add these together, and if something ends up on the left and the right side of the reaction, then it cancels out. So these species that are formed in one step, the hydroiodic acid, and then consumed in the next step, those are reaction intermediates. So they occur only, well, usually temporarily before being converted into the final products. So the reaction mechanism gives you a complete detailed description of what's happening at the molecular level. So like step by step, this is actually what's occurring instead of just looking at what goes in one end and what comes out the other. It's kind of like a factory, right? If you had a factory and they're shipping in, I don't know, aluminum and different metals and like plastic pellets into a car factory, and then cars come out the other end, the overall reaction mechanism is metal and plastic in, car out. The individual steps are the reaction mechanism. So the metal comes in, it gets cut into the right shapes, it gets formed, the plastic gets melted down. There's all these steps that take place in between, but we only see the final product on the outside. So this is kind of opening up the black box, looking inside. How does this chemical factory work? So we can use chemical kinetics to determine the mechanism of a reaction, or to at least help try to determine the mechanism. So the way you do it is you start back, or you work backwards, and you write a mechanism, or you measure the kinetics of the overall reaction, and then you work backwards to write a mechanism consistent with those kinetics. So we would take, actually, let's use this one as an example. So if we want to know more about how this reaction works, we would take hydrogen, and we would take iodine monochloride, we would put them into a reactor, and we would do what, like initial rates, right? So we would put in known concentrations, and then we'd vary those concentrations, figure out what the rate law is, calculate what K is, and then we know that this is the actual rate law. This is actually what we've observed with our experiment. Now let's try and work backwards from that information and figure out what steps could have caused that rate. One of the first things that we can do is we can use this idea of molecularity. So talked about for overall reaction rates, or reaction laws, or sorry, overall rate laws, you have to use experiments to determine what the rate law is going to be. We can't look at the reaction and say it's going to be second order for iodine monochloride and first order for hydrogen. We have to actually do the experiment. For the elementary reactions that we're going to be creating, you can use, it's called molecularity, to determine what the rate order for that elementary step is going to be. So it becomes pretty simple here. We've got unimolecular. So A turns into products. So that would be first order. Actually, I think we get to this. Yeah, cool. I'll get to the corresponding orders, but I think they'll make sense. If there's bimolecular, it could be A reacting with another A molecule to form a product. Or we could have a different bimolecular where A is reacting with something else to form a product. So bimolecular, 2 molecules involved. You can have termolecular steps, but remember collision theory, the odds of getting 3 separate molecules to collide, let alone with the right orientation, is going to be very, very rare. It's not going to happen very often, so termolecular steps are going to be incredibly rare. And for this class, when we do, because we'll have a quiz on this, Think either bimolecular or unimolecular. So the rate laws for elementary steps can be deduced from their equations. So if we have a unimolecular step, zoom this a little bit. If we have a unimolecular step, molecularity is 1. And so our rate law for that unimolecular step will be first order with respect to A. And then since that's the only molecule involved, that would be our rate law for that elementary step. At this point, hypothetical, theoretical step. If we have A reacting with another molecule of A, our molecularity is 2 molecules involved. So it would be bimolecular. Then the rate law for that elementary step is going to be K times A to the second. So second order with respect to A. And then following from that, if we have A reacting with B, again, it's bimolecular, but now it's 2 different molecules. Still has a molecularity of 2, K times A times B. So overall, second order, but first order with respect to each of the reactants. And then we could have termolecular, and that could be, I guess it would be, is it 3 times 3 different combinations? Might be 3 factorial. So I think there'd be 6 total combinations, right? Because it could be AAA, or it could be AAB, or it could be ABC, or it could be BBB, right? There's a lot more combinations that are possible. But these termolecular steps are going to be rare. But the rate laws for them follow the same pattern. Right? If it takes 2 A's and 1 B, then it's going to be second order with respect to A, first order with respect to B. Right? You just add them together. So usually, it's one step that's slow. So you've got a factory line, your first, maybe it's the first spot in the factory line that's really slow. That's the one that's backed up. Everybody else after that is waiting. So let's say we're in the factory and you've got sheet metal and you're pressing it into the right shape. For whatever reason, that takes a long time. So everybody after you could go a lot faster if you could press more sheet metal. Or in this case, if you have a car accident or if they're paving a new road, everybody has to funnel through this choke point. So that is the slow step is the rate determining step, because it can't go any faster than that choke point, than that slow person on the factory line. And so we'll be looking for the slow step and sort of zeroing in on that for determining what the overall reaction rate is going to be. All right. So if we have nitrogen dioxide, carbon monoxide, we know it reacts to form nitrogen monoxide and carbon dioxide. The first step is going to be to figure out what the overall rate law is. So for this overall chemical reaction, what's the rate law? And how can we do that? Oh, well, we can't use the molecularity yet. That's only for elementary steps. We can find K. We have to find our reaction order first. So what experiments do we have that we could find the reaction order with respect to each of our reactants? Huh? A graph? What would we graph? We could do initial rates. Yeah. So initial rates is kind of what I was looking for here. We could graph with changing concentration for each one. Or we can do an initial rates experiment. So we would put in, these are gases. So we can use atmospheres interchangeably with molarity because the concentration is directly tied to the pressure of the gas. So we could put in like 1 atmosphere of nitrogen, 1 atmosphere of nitrogen dioxide, 1 atmosphere of carbon dioxide, measure what the rate is, and then maybe we double our carbon monoxide, right? So we can determine what that overall rate is. There we go. So we would get an experimentally determined rate law. And then for this reaction, it would be this. So the rate equals K times the concentration of nitrogen dioxide squared. So what is something that you notice about this? How does this relate to our overall reaction? What? Oh, go ahead. It's fine.
Yeah, the biomolecular.
Oh, bimolecular?
Yeah, the biomolecular.
Yeah. You're kind of getting ahead of where I was thinking, but yes.
Because it has the two.
Yeah, so we're going to be looking for a slow step that's going to be bimolecular with respect to nitrogen dioxide. The other thing I was looking for is our rate doesn't depend on the concentration of carbon monoxide. It's not present in that rate. So changing that rate, zero order, not going to matter. So our rate limiting step is going to be bimolecular with respect to nitrogen dioxide, and it's going to be, it's not going to have carbon monoxide in it. So could this be a single step reaction? It can't, right? If it was a single step reaction, then our only elementary step would be this one. But there's only one nitrogen dioxide here, and we have carbon monoxide. So this, just as a hypothetical here, I'll put in quotes, air quotes. If this was hypothetically a single step reaction, then the rate for that elementary reaction would be K times NO2 times carbon monoxide, right? That would be the rate law if it was single step. And since we experimentally determined what that rate law is, we can look at this and say, no, it can't be single step. And as we've already discussed, it's also told us some stuff about what our rate limiting step is going to look like. So one possible mechanism would be this, that we have, and we'll go through more of these, of course, but possible mechanism might look like this, right? So first step is a slow step, and it's got to be bimolecular with respect to nitrogen dioxide. So nitrogen dioxide is reacting with nitrogen dioxide, and we're getting nitrogen trioxide and nitrogen monoxide. Then in a second step, the nitrogen trioxide then reacts with carbon monoxide to give us nitrogen dioxide and carbon dioxide. Yeah, nitrogen dioxide, carbon dioxide. So you might be thinking this last step here doesn't have nitrogen monoxide, and that needs to be one of our products. Well, let's go through this. And nitrogen trioxide is on both sides. And then we have two nitrogen dioxides, so we can take one of these and it'll cancel out. Actually, maybe let me use a different color. Right, so since we have nitrogen dioxide on both sides, is that hard to see? The pink is not good. All right, I'll switch to something else. Here, I have yellow. So we got nitrogen dioxide is on both sides, and we have two in the reactants and one of the products, so one of them is going to cancel out, and so will the nitrogen trioxide, so that when we combine these two steps together, when we really are just adding them together, this will be NO2 plus CO, make nitrogen monoxide plus carbon dioxide. Right? So that matches with our overall reaction. And because our first step is the slow step, that's the one that's going to determine the rate. Any Any questions so far? Nope. All right. So we've looked at some energy reaction diagrams, and I know you've had them on Alex. The first step for this reaction in this energy reaction diagram has a higher activation energy. I do want to point out that the activation energy is the energy from the reactants up to the transition state. So it's not just from the floor up to the transition state, it's from wherever the reactants start at. And then for the second step, right, so this goes up into a transition state. After it's past that transition state, it comes down to a second energy level, so that transition state has, or that intermediate, has a higher energy than the reactants. And so the energy of this transition state is from that floor to the peak of the next transition. So because this energy is higher, right, there's less likely to be molecules that have enough energy to get over that barrier. That's going to be the rate limiting step. So the step that requires more energy is slower. So our first step determines the overall rate, and then anything that makes it over that first hurdle can pretty quickly go into the next one and down into the products. And then the other thing that gets thrown in here that we will talk about later is this delta H of reaction. So that's how much heat, actually I think you should have covered this at some point. That's the heat that's produced by the reaction in this case, right? Because our energy level of our products is lower than our reactants. So this is going to be generating heat. It'll be an exothermic reaction. So we're trying to come up with valid mechanisms to explain chemical reactions. There are a couple conditions that need to be met. The first one is that the elementary steps in the mechanism have to sum or add up to the overall reaction, right? We can't put extra steps and extra chemicals in that, or compounds that don't cancel out, because then we're describing a completely different reaction. You can really kind of put in whatever you want as long as it cancels out, and as long as it makes sense at the end for what the final product is. And then the rate law, the second condition is that the rate law predicted by the mechanism must be consistent with the experimentally observed rate law. So it has to match what we see in our experiment. Because if it doesn't match what we see in our experiment, then it can't be the correct mechanism. Which is why that's where you have to start, is with an experiment determining that rate law. And mechanisms cannot be proven, only validated, because we can't actually watch the atoms or molecules react to see if they follow the steps that we think that they do. If an intermediate exists long enough, you can prove the existence of an intermediate. Or maybe if you've got a rate law, you can sort of predict what the rate of an intermediate converting into your final product would be. If you can create that intermediate by a different manner and prevent it from going to the product. But those are really only validations and not proofs. Because unless you can show something from an intermediate or something, there might be other mechanisms too that can fill that same reaction or that can match the experimental rate law. Okay, yeah, we're gonna go through a couple more examples. So that's where we have a reaction with a slow initial step. We can run into a weird situation, a little bit of a weird situation, where the rate limiting step is not the first step. So if the slow step is the second step or the third step, then you can end up with an equilibrium between your reactants and whatever product they make in that first step. And then if the next step is the slow step, then that intermediate will exist in a substantial quantity and only be slowly used up by the slow step. So you guys remember the concept of equilibrium? This is where, the rate of the forward reaction is equal to the rate of the reverse reaction. So that's kind of what we're talking about. So here's one example of this. If we have hydrogen reacting with nitrogen monoxide to produce water and nitrogen gas, this first step is fast. So the nitrogen monoxide quickly reacts with another nitrogen monoxide to produce dinitrogen dioxide. And because that happens so fast, it builds up enough of a concentration that the reverse reaction has a substantial rate. And so the rate of that forward reaction will equal the rate of the reverse reaction, and only be used up by this second slow step, where the hydrogen slowly reacts with the dinitrogen dioxide. Then the step after that's fast. So we can show that this mechanism adds up to our overall reaction. So we've got, let's see, dinitrogen dioxide on both sides, dinitrogen monoxide. Yeah, and then we can add this up, and it'll match the one on top. 2H2 plus 2NO reacting to form 2H2O plus N2. But our slow step is this one in the middle. So based on that slow step, what would our rate law be?
The rate equals K times the concentration of H.
Yeah, hydrogen
H2
times the N2O2. So that would be the rate law for that one slow step. But that doesn't match the rate law that we determined for our experiment. So because we can't tell if this is actually what's happening in the reaction, we can't just use this. We have to sort of adapt it to what the overall reaction is. So we need to take this amount of N2O2, the dinitrogen dioxide, and express it in terms of the reactants that we have in our overall reaction. And sometimes this is easier, sometimes it's harder. For this one, we can say that, well, we can kind of do this just by observation, just by inspection. And we can say that the amount of N2O2 is going to be directly tied to the rate that nitrogen monoxide is converted into it. So we can write that rate law as K times the concentration of, NO squared. And that matches with our reaction, or the overall experimentally determined rate law. The other way to do it, though, is with a little bit of algebra. So we can express, this is getting a little bit into the next chapter, we can express this as, I'm going to zoom in here. So that's 2NO. So the rate of this forward elementary step, what would be the rate law for that? Call it rate equals for that one step. Yep, times the concentration of NO squared. So we'll call this rate 1, and this is K1. What would the rate of the reverse reaction be? How would we express that rate law? We'll call it rate -1, because it's going in the opposite direction. K -1. Yeah, concentration of N2O2. So now we want to express And then we need the rate law of our slow step. So rate, call this one rate 2, which is the rate of step 2. It's going to be K. And then what would the rate of step 2 be? Concentration of H2. Oh, wait. I don't want to. This is step 2. H2 times the concentration of N2O2. So we want to now replace this N2O2, which is an intermediate, and it's really only a hypothetical intermediate in our proposed mechanism, with a rate that describes that. So we say, well, the rate of this forward reaction and the rate of this reverse reaction are going to be at equilibrium, because that stuff's fast. So it'll produce enough N2O2 that it's going to be converting back into NO. And so we can say set those equal to each other. So K1 NO squared equals K negative 1 N2O2. So then the concentration of N2O2, we just move K negative 1 over, right, divide both sides. And now we can say that K1 times NO squared over K negative 1 is equal to the concentration of N2O2. And then we substitute this in for N2O2. And actually, I did miss something up here, that the hydrogen is also part of that. All right, I'll zoom out again. So now, rate, that's K2. Rate is equal to K2 times the H2 concentration times K1 over K negative 1 times concentration of NO squared. So we can collapse that down a little bit and say that the rate of K2 times K1 over K negative 1 is H2 times NO squared. So now this matches our overall reaction, and so the K of the overall rate law must be equal to K2 times K1 over K negative 1. I know that last bit is a little bit hand wavy, but we're saying that if this is the correct mechanism, then the K value is actually made up of the forward K of rate of reaction, or of step 2. It's the forward K of step 2 times the forward K of step 1 divided by the reverse step 1. So the overall K value is actually made up of these pieces. Are there any questions about that? We're going to do another one. Actually, what we're going to do, because I think we're going to have plenty of time, is I'm going to let you guys work on the next one, talk with each other, and then we'll meet back up in a few minutes to talk about it. Because this is the kind of problem where you really do need to use the equation to figure out what it is. Because there's oxygen to the negative 1. Okay. should have been enough time to get started. So what's the first thing that we can do to show that this is a valid mechanism? So the first condition is showing that the steps add together. So if we look at our mechanism, proposed mechanism versus the observed mechanism, or the observed reaction, we've got 2O3, this oxygen. Those are on opposite sides of the reaction arrow, so those will cancel out. And then we've got 3 oxygens on the product side. So that should add together to equal the reaction mechanism. And then for the rate law, how do we get this rate law in this format? Because our first step here is fast, right? So the forward reaction there, that would just be dependent on, it would be first order with respect to ozone. So we need to incorporate this equilibrium with the reactants in the second slow step, second comma slow step. So what equation would have set up first?
The rate law for the forward reaction in the first step?
Yeah. Rate of the first one, so rate 1, it's going to be K1, and then concentration of ozone. And then what's next?
The reverse?
Yeah, rate of the reverse. So the rate of the reverse is going to be equal to, so it's rate -1, K -1. That'll be the concentration of oxygen times the concentration, well, dioxygen versus oxygen. And then the rate law for our second step. So for the second step, rate 2, it's going to be K2 times O3 times atomic oxygen. Now we want to replace the atomic oxygen, right? Because that's our intermediate. That doesn't show up in the experimentally determined rate law. So if we set the rate of the forward, step 1, equal to the rate of the reverse, step 1, and we can do that again because they're at equilibrium, right? The products of that first reaction aren't being used up fast enough, so they come to a balance, and the rate of the forward is going to be equal to the rate of the reverse. So that'll be K1, or actually, wait. Just want to make sure I have enough space. Yeah, we can do it up here. K1 times ozone equals K -1 times molecular oxygen and atomic oxygen. And then rearrange so that we just have, or rearrange so that it's equal to the concentration of atomic oxygen. It should be K1 over K -1. Ozone on top, oxygen, molecular oxygen on the bottom. That'll equal the concentration of atomic oxygen. And then we just replace that in our rate 2. Sorry. So rate 2 is going to equal K2 times O3. We replace that monoatomic oxygen with K1 times O3 over K -1 times molecular oxygen. And then when we bring all of those together, then rate is equal to K2, bigger, K2 over K1, or sorry, K2 times K1 over K to the negative 1. O3 squared over, or actually, let me write it so it matches, O2 to the negative 1. So now if K for our experimental is equal to this K2, K is equal to K2 times K1 divided by K negative 1, then our rate law determined here for our elementary steps is equal or the same as that of the observed experimental rate law. So really what we're doing is we're checking, with our mechanism as proposed, can we write a rate law that matches what we observe experimentally? Even if that means that there's a weird oxygen to the -1. Does anybody know why oxygen is to the -1 here? So if this first step is in equilibrium, remember Le Chatelier's principle? If we add oxygen, if we were to add more oxygen to this reaction, it'll push the equilibrium of that first fast step back to ozone. So by adding oxygen, it slows down our fast step, which is feeding into the slow step, and would have a negative effect on the rate. Cool. Now, the harder part is predicting what the mechanism might be. So here we've got, well, actually, here we're given a mechanism. Do we do one? We don't do one where we predict the mechanism. Okay. Just for some reference for like an exam, I would give you the experimental rate law, and that gives you a lot of hints as to what the mechanism is going to look like, because it tells you what your slow step is going to be. And then I'll tell you something like, and I think Alex does this way too, tells you which step is the slow step, and if it's going to be bimolecular or unimolecular. So then you kind of know what pieces need to be placed where. But for this one, let's predict an overall reaction and rate law that would result from the following two-step mechanism. So first thing to check, is the slow step the first step, or is it a different step? The first step is the slow step. So that makes it easier, because that means that this is going to be the rate limiting step. So we can write our rate law based on that slow step and not have to do all of the math and equal rates and equilibrium stuff. So what's this rate going to look like? I guess that's part of this, right? That's our rate law. Yeah, and so because that first step is the slow one, we know that anything that comes out of this first step is going to be immediately used up by the second step. So it's going to have no bearing on the overall rate, how we get from our reactants to our final products. And then the overall reaction would be 2A, and then A is on either A2 is on either side of the reaction arrows, so that'll cancel, and it'll be 2A plus B forms A2B. Cool. Any questions about reaction mechanisms? All right. So the last thing we're going to talk about, and it's not going to involve any math, is catalysis. So when you catalyze a reaction, what's happening? I think we mentioned it a little bit before, but the rates of chemical reactions can be controlled by, well, we know this. We can alter concentration or temperature, but there are limits to that. Or you can only turn up a reaction temperature so high before you either start boiling it, things start reacting with air or oxygen, or your reaction vessels can't handle the temperature, problems with concentration. If you increase the concentrations of things too high, maybe a little bit of nitric acid for your reaction is fine, but if you add too much, it starts to dissolve your reaction vessel, or it's too expensive. And so the much, much easier thing to use, or thing to do, is use a catalyst. So instead of it throwing in lots of material, turning the temperature way up, which the other thing that can happen is if you turn the temperature up too high, is now you have a lot of energy, and you can have unwanted reactions take place. Because now any activation energy barrier that you couldn't cross at a normal reaction temperature, you'll be able to make side products and junk. But adding a catalyst increases the rate of the reaction and doesn't use up the catalyst. So by definition, a catalyst doesn't actually become one of the products. It just lends a helping hand by providing an alternate mechanism in which the rate determining step has lower activation energy. So this is something that if you know or you have a proposed mechanism for intermediates, maybe you can add that intermediate in, or maybe you can add something that is generating that intermediate by a different step, and so you can sort of tinker with your reaction on a more detailed level. And catalysts kind of do that by offering a different pathway. So energy reaction diagram again. For the original reaction that's uncatalyzed, that's this blue, the blue path I drew on with the red highlighter. That activation is high, so it's slow, but in the atmosphere, the decomposition of ozone gets catalyzed by chlorine. Chlorine offers a different pathway. So this first reaction happens with much lower activation energy, and then it gets pushed along to the products. Here's the mechanism for catalytic destruction of ozone. You see that chlorine is one of the reactants. Chlorine is also one of the products. So in this first step, you form this hypochlorite. I think it would be hypochlorite. Plus oxygen. Then that hypochlorite goes and finds another oxygen and creates, or atomic oxygen makes a molecular oxygen, and the chlorine gets released again. So it's a lot easier for ozone to react with chlorine than ozone to react with atomic oxygen that might be around. And so because that first step happens more easily, it happens a lot faster. This is actually why you can't actually buy Freon anymore. And a lot of chlorofluorocarbons, fluorine, chlorine, bromine, all catalyze the destruction of ozone. I actually know a ton about this because one of the chemicals that I worked with is a post-harvest fumigant. Use it to kill bugs when you're shipping fruit mostly. Is CH3Br, methyl bromide. And that bromine in the methyl bromide can get up into the upper atmosphere. It gets cleaved off of the methyl bromide by light photolysis. And then you have bromine floating around, and it reacts with oxygen in a very similar catalytic pathway. So there was this global effort called the Montreal Protocol to eliminate these kinds of compounds. Now you can still get them. Actually, if you've ever bought like Dust Off, right, that can of air, compressed air, it has fluorinated carbon compounds in it. Fluorinated hydrocarbons. And they can do this same thing. We've come a long way, and the ozone layer has recovered. But we'll talk more about ozone. The other one, this one that you may have been aware of or not, catalytic converters in your car. Not required in every state, but in California, you're required to have a catalytic converter, and that's because the rhodium or platinum, rhodium, palladium on high surface area substrates take exhaust pollutants and converts them into less harmful compounds. They can also take unburned fuel fragments and convert those into CO2 and water, so we don't have these hydrocarbons floating around after the reaction. And so the reason that people will steal these is because you can sell them to have the platinum or the rhodium or the palladium removed, which are rare metals and very expensive. So two types of catalysis: a homogeneous catalysis, where you've got the catalyst in the same medium as your reaction. So for the chlorine in the upper atmosphere, right, that's a chlorine gas reacting with ozone. That would be a homogeneous catalyst, or the process is homogeneous catalysis. Heterogeneous catalysis is like the catalytic converter. It's a solid matrix that has platinum or palladium in it, and air, exhaust, passing over it reacts. So one's a solid, one's a gas. Here's that ozone story. So ozone depletion gets worse depending on the time of year. So heterogeneous catalysis is most likely responsible for the annual formation of an ozone hole over Antarctica. But chlorine is evenly distributed throughout the entire atmosphere. And the reason this hole forms over Antarctica is that the chlorine's actually trapped in these, I looked up what the name of these was. It's like chloronitrate compounds. And so the chloronitrate isn't able to do the catalysis and destroy ozone. However, when you get into the winter, Southern Hemisphere winter, right? So it would be our summers. Like right now, the ozone hole over Antarctica would be pretty bad, or worse at least. The ice particles in the clouds catalyze reactions that release the chlorine. So that chloronitrate has trapped all of the chlorine, but then it can be catalyzed by ice in the clouds, releases that chlorine, and now the chlorine is able to be photolysed, undergo photolysis into atomic chlorine, where then it can catalyze the destruction of ozone. So because it gets colder, there's more ice clouds, or more ice in the clouds, more chlorine gets released, more ozone's destroyed. So it's a little seasonal. So there's double catalysis happening there. If you do go on to take organic chemistry, you might get to do this reaction. I don't know. This is a common reaction in organic chemistry, though, where you take double bonds between carbons and you use a palladium, platinum, or nickel catalyst. And it's a solid catalyst. And what it does is it adds hydrogen to that double bond, makes it a single bond. So what it does is you actually get your carbon-carbon double bond, in this case ethylene, gets adsorbed. I don't know if you know the difference between absorb, AB. So there's absorb. Absorb is when it gets actually sucked into it. So like a sponge is absorbs things. Your hair actually doesn't absorb water, it adsorbs it. So it sticks to the outside of your hair follicles. So same thing is happening here with the ethylene. The ethylene sticks to the surface, gets held in place there for a little bit. Reactants are adsorbed onto the metal surface. Reactants diffuse on the surface until they approach each other. So you've got ethylene here stuck to the surface, and you've got hydrogens all moving around on top of the surface. Then when the ethylene comes into contact with a hydrogen, it reacts, and the catalyst delocalizes that double bond a little bit. And so then the hydrogen can add into there without having to fully cleave the double bond. And then the last thing we're going to talk about here for catalysis is biological catalysts. And I think this is what I mentioned something about. Enzymes, though, are really cool biological catalysts. They're highly, highly selective. And they're usually large protein molecules. And their goal is to lower the activation energy of cleaving certain molecules or putting molecules together. And it does that by grabbing them and then straining bonds, or it'll grab two separate things and kind of smash them together in the correct orientation. And they greatly lower the activation energy of the reaction. So the general binding mechanism is this. E here is your enzyme. S is the substrate. So you've got your enzyme and your substrate. They bind together really fast. Then the slow step is the actual process of breaking the bond or forming a bond. In this case, I think this is for, it's sucrose. So you've got a big sucrose molecule. It'll be on the next page too. It fits into that binding site, and then that sucrase is what it's called. Strains the bond between, because sucrose is made up of fructose and glucose, it strains the bond between the two. So then when that bond breaks, it then lets go of this glucose and the fructose. And there's a particular enzyme that's like animals that eat like grass and stuff, they can digest I cannot remember what it is. Cellulose, though, is, I think cellulose is glucose, but it's one of the bonds between the glucose and the fructose is flipped in the opposite direction. And that happens to matter a lot. So since they're orientated differently with respect to each other, our bodies can't cleave that bond and turn that sugar into something that we can use. So we lack the enzyme that fits that molecule, and so we can't digest it. Other animals or bacteria in their gut have that molecule. So sucrase, that ASE, means to cleave. It's it's an enzyme that cuts things. Sucrase, lipase cleaves lipids, which are fats. But it grabs onto them, strains the bond between the two. Zoom in on that. So this bond right here between this, what would be glucose, and this, what would be fructose, and it strains that bond so that they can be cleaved into glucose and fructose. And then, of course, this is drawn as a huge blob because it's a giant, complicated protein. One more aside about that. These proteins are really cool and they're really complicated. And we know what a lot of proteins, at least for the most part, are made of, what composes them, how much nitrogen, oxygen, carbon, that kind of thing. But if we try to put them together, we don't know why they fold the way that they do. So there's this project called the Folding at Home, used to be called Fold It, where they're using people's computers. You download this piece of software, and while you're not using your computer, it computes different folding patterns to find low energy states for those proteins. And if we can do that, figure out why proteins fold the way they do, things like, mad cow's disease is one, where there's misfolded proteins. There are other misfolded protein diseases. Anyways, we can learn about them. how to make them, how to combat them, or how to mimic what they do. So like if we could make sucrase, if we can make other enzymes to make more complicated molecules, be a huge breakthrough. So enzymes give organisms tremendous control over which reactions occur and when they occur, how fast, how slow. Like I said, enzymes are extremely specific, and they only catalyze each a single reaction. So they're all designed to do a single thing. Enzymes speed up reaction rates by factors as large as 1 billion. So happening 1 billion times faster than it would otherwise. And then many substances that inhibit enzymes are highly toxic because they're stopping these processes that don't happen otherwise. So locking up even just a single enzyme molecule can prevent the reaction of billions of substrate molecules. Which actually, that was something that was also part of my research. I had reviewers ask about the toxic mode of action for the fumigants in my dissertation. And they really come down to, oh, I wish I thought of this earlier. One of them stops glycolysis. So part of the, have you guys taken biology? Yeah, okay, most people, right? So glycolysis is like the first step of the Krebs cycle, basically. One of my fumigants that I used stops glycolysis. And then that produced an effect in the insects where they could live for a while because insects, just like us, have other ways of sort of feeding the Krebs cycle to generate energy. One of those being breaking down proteins instead of sugars. So the insects could digest sugars for a while but couldn't keep up with their own metabolism and would eventually die. The other one stopped cytochrome C oxidase, which is another enzyme. And it's the last step of the electron transport chain. So it's the very, very last step that creates energy. I'm not a biologist or an entomologist, but I had to learn some of it. But because it stops that very last step, there's nothing to replace it. That enzyme is the only one in these insects that does the last step of the Krebs cycle to create that energy. And so because they had no cellular respiration, they died a lot faster because of that fumigant. And that's blocking a single enzyme for both of those. They're invasive species. They're a problem. Okay.